Thank you.
Okay. So, you know, and whenever we have a transfer like this, right, and we have a table like this, of course, you should be familiar with how to, you know, how to write the equations, write them one way, turn them around, write the other way, add up your E zeros, make it come out positive, get your delta G from minus NF delta E, right, and know what we're talking about when we talk about this reduces that, and this becomes oxidized. And this. So that languaging can be confusing sometimes, but it's essential that you get more familiar with it. Because that's this whole aspect of the class is basically about this kind of stuff. Okay, so the electron transfer chain in mitochondria consists of 5 protein complexes. If we just look at the electron transfer chain itself. Really, it consists of 4 complexes. So let's just say it's 4, 1, 2, 3, and 4. These complexes have specific names associated with them that we'll get to. I don't care if you remember those names. I'm just going to call them complexes 1, 2, 3, and 4. The names are impossible to remember, except for maybe number 2. So, I mean, here are some of these are kind of simpler names, you know. But so what can you do? This tells you something like this is how big it is. You know, this one's almost 1 million. The other ones are somewhat smaller. Right? Cytochrome C is an important one. Right? It's not really an enzyme complex. It's written separately to indicate that it has a special function on its own and not part of one of the 4 complexes. So you've got the 4 complexes and cytochrome C and ATP synthase and some associated proteins. Right? So how many subunits? It's big and complicated. That's all we need to take away from this. And this is really a partial list of some of the cofactors, the redox cofactors that are bound on these complexes. Okay, so what you can do is you can isolate mitochondria from cells and slough away the outer membrane and then disrupt it with some kind of detergent, like digitonin is a detergent. Get rid of the outer membrane fragments. Take fragments of inner membrane and reconstitute them in little vesicles as little detergent-soluble fractions. Just like you're purifying a protein and you run an anion exchange column and a gel filtration column for size or whatever, you end up with fractionated proteins. You can fractionate complexes in the mitochondrial membrane and separate out the 4 complexes and ATP synthase. Okay? And then you can feed them with redox cofactors and see if, just in vitro in the test tube, and see what they do. Right? So we know that we can isolate them, and we know we have them separate. We know, for example, that complex 1, if we feed it NADH, reducing power, we'll get reduced Q out of it at the end. And it'll kind of be a one-shot reaction. We can feed it NADH, we get Q out. That's all it can do because we have isolated that complex, but we know what it does now. Right? We know how to get it. Similarly, complex 2, succinate comes in, and reduced Q comes out. Complex 3, reduced Q goes in, and reduced cytochrome C comes out. So electron transfer from Q to cytochrome C. And then complex 4 from cytochrome C to oxygen. Right? And then here's the ATP synthase fraction. Is it synthesizing ATP? No, it's hydrolyzing ATP. Why is that? Has no gradient. There's no intact mitochondria. There's no proton power to harness to drive ATP synthesis. So the enzyme's there, it's intact, right? But if you feed it ADP and phosphate and say, hey, where's my ATP? Of course, nothing's going to happen because there's nothing to power it, right? What's interesting, perhaps, is that if you give it ADP and phosphate, if you give it ATP, it'll happily hydrolyze it. So there's an active site there that works, it's just working the other way. So we'll come back to that when we talk about how that enzyme works in detail on Wednesday. Okay, so the chain functions in 4 membrane-bound supramolecular complexes, meaning that there's a lot of protein in them. And we can fractionate them and study them individually, and that's how we know what each one does. All right, so this is the most important slide. So every detail on this slide is important and gives you the big picture of what's going on. So we're going to talk about some details of each complex, but it's much more important that you get the big picture from this slide than that you remember, lots of details from the other slides. So what's going on here is NADH is delivering electrons. And by the way, I noticed that there's a figure in the book, in the later version of the book, version 6, where instead of NAD, this arrow goes the other way, so it's a bad typo. It has NAD going to NADH instead of the other way around. This is correct, huh? So we're delivering electrons here in the form of NADH from TCA cycle, say, or in the form of succinate, okay, which is also TCA cycle. In fact, complex 2, we mentioned before, is succinate dehydrogenase of the TCA cycle. So complex 2 up there is just another, is a TCA cycle enzyme that we already talked about. And all it does is take succinate and turn it into fumarate. Right? That's not all it does. It turns succinate into fumarate and takes electrons from succinate and funnels them through its own protein structure. Right? So the little blue lines here indicate electron transfer. Okay? So electrons are transferred from NADH up to Q, which you've seen the structure now of coenzyme Q. Q becomes reduced Q. Right? Notice here that Q is diffusing in the membrane, this little dotted black line. It just indicates that QH2, reduced Q, is diffusible in the membrane. This is why it's important that it be diffusible. Right? So this is just an artifact of the artwork, right? It just to show that. It doesn't go out of the membrane, stays in the membrane, just gets around complex 2. So succinate's electrons also end up in QH2. So the reduced Q from both complexes 1 and 2 ends up at complex 3. Okay. Then we have more electron transfer events at complex 3, also involving Q. But ultimately here, Q gives up electrons to cytochrome C, which becomes reduced. And in cytochrome C, a 13 kilodalton soluble intermembrane space diffusible protein drifts off from complex 3 to complex 4. Okay. So there it goes. That's the drift. So it arrives here, and then from cytochrome C, electrons in blue again are transferred to oxygen to form water. So the other thing that's going on here, right, is to look at the red, or the pink, and to note that these electron transfer events are coupled to proton transfers. Right? So here we've got protons coming up from the matrix, right, going into Q. We've also got protons just shooting right through. Right? 4 protons shooting right through complex 1, going from here to here. And these numbers are per pair of electrons delivered by NADH. So this makes the intermembrane space more positively charged. Complex 2 has no proton transfer. Right? Very important to note this. Complex 2 has no proton transfer. Right? Which cofactor in succinate is delivering electrons? FADH2. Succinate dehydrogenase. So FADH2 does not contribute to setting up proton gradient on the complex it arrives at, whereas NADH does. Right?
Can you say that again?
NADH arrives here, transfers electrons through complex 1 to make QH2. In the process of doing that, it is able to couple through the structure of complex 1 to get protons ejected into the intermembrane space. Whereas when succinate here is oxidized to fumarate and FADH2 forms and then gives off electrons up to Q. It forms Q in the end just as this guy does, but there's no red arrow going through, which means that the capacity of FADH2 to generate a proton gradient is less. Even though they both generate QH2 and the QH2 is going to itself help generate a proton gradient, so it's not like FADH2 has no capacity. It does, because when it generates QH2, it's going to have capacity here and here. Right? But it just has less. In fact, it's got 4 plus 2 is 6. 4 plus 4 plus 2 is 10. So that's the difference between FAD and an ADH2. Sorry, FADH